Welcome again to day two. So this morning before coffee, we're going to do again another session. This is session 3. How does everything interact? Quite a lot to get through. This is quite a number of lectures compressed into one. And again, go back to the YouTube if you want to have a look at things in more detail, or if you request, we'll do a detailed session in February. Okay, so we're going to have a look at how do things interact. Well, let's start about this quote from our good Nobel Prize winner, Richard Henderson. And, you know, we can kind of invent new ways to deceive ourselves. So when you come to doing assay development, you don't want to deceive yourself. Here's a case in point. This is a functional MRI of a salmon, and it's responding then to pictures of human emotion. And look, in the brain, there's kind of these lighting up spots. This is quite amazing, isn't it? Right? There's only one problem. It was dead. And so what this was actually published in this exciting journal called Reproductivity on expected results. And they did this just to demonstrate the fact that if you don't take away your background, you can get these false positives. And, you know, in the case, you know, of this dead salmon. So the thing you want to do is make sure you don't have a dead salmon as a project. So you're going to think first, you're going to think about what your biological model is. How are you going to interpret that? What methods of controls you're going to have a look? You're going to then look at, you know, is this reproducible, what you can actually see? You know, do you have valid targets? Are you looking at this process with something that's actually valid? You're going to develop a physical model. And the reason I was going to say, and so, you know, reproducibility is a real crucial thing at the minute. Lots of drug discoveries kind of fail in the first kind of trials, and that's because really about this reproducibility problem. It was such a problem that these companies actually went back to actually test kind of landmark papers. You know, Bayer Healthcare went through 67 academic published targets. They could only validate 25. So the over 75%, if they went drug discovery with those, it would have been completely invalid. Amgen then went through 53 studies, landmark studies, only could confirm 11. And the cancer reproducibility only could produce 46% of 112. So the crucial thing is that a lot of literature out there, it may not be true. So just be cautious. Also, in terms of reproducibility now, the journals expect that you're going to show how many times you've done things. You're going to have to deposit your raw data, and you're going to have to be very clear about how much you reproduce things. Okay, so what are the roadblocks to reproducibility? Obviously, it's a big push to publish. You know, so let's just get that done. Let's get the paper and don't forget about the controls. Let's get that done. Also, it can be cognitive bias. So you can kind of think, well, I'm only going to just pick the result. Or you can think, yeah, I know I'm right. So this result is wrong because it's not confirming what I know is true. Other way is selection bias. You've done a whole range of experiments and you've done it 10 times. It's only one that worked once. And you're going to go, oh, well, it must have worked. There we go. They only select the positive ones. A bandwagon where basically a field can be taken over by a certain viewpoint and actually, or a set of results. I mean, there's kind of a famous case if you want to have a look up about the alpha beta 50, I think it's a 52, 56 in Alzheimer's. And yeah, it's a whole barrel of wrong direction, really, because of possibly even falsified results. Okay, and cluster illusion is kind of just seeing kind of patterns in the noise kind of thing. As humans, we're very, very good at looking at patterns and drawing kind of dots between them. And such like in say like in Victorian times, people were looking at Mars and went, oh, because the resolution wasn't good enough, they could see these kind of canals. They thought were canals, but obviously there's not canals on Mars. Right, reporting bias. Obviously, in the literature, everything is reported as positive. It's very, very difficult to look at stuff that's actually negative. And of course, poor methodology, which hopefully we're going to counter in these lectures. Right, and again, you haven't done it enough times, and so you've got to lack statistical power. Okay, so what's this experiment going to tell me? I'm going to do a physical model. What kind of signal am I going to look at this? You know, what are the assumptions in that? Is it representative of the process? You know, what's the raw data? You know, what this is precision and accuracy? And of course, what we mean by precision means that you take a measurement and it's going to be the same the whole time. But it could be, say, completely wrong. This could be where the actual point is of the real data point. But you've been very precise, but you're wrong. Obviously, accuracy, really means that you will eventually get that average value, even though you might have kind of random noise around the value itself. Okay, what controls are you going to do? Are they good enough? When you correct the data, have you done it for enough time? You've got enough concentration range? Have you let things equilibrate for long enough in kinetic data? Or you have got enough time to actually look at that process and fit it accurately? Okay, you're going to have some kind of data model, and then you're going to fit. So what should a good assay really kind of look like? So here's a high throughput screen I did done with Mark Stubbs in the ICR way back when. And what we're kind of looking for is really a good signal window here. So we can say, this is bound and this is bound and this is unbound. And we're going to relate that against what the noise is. And this is the stigma factor here. So you can see that these are all various batches of protein, different high throughput screens. And when we started off, and you can see when we started off, it wasn't very good. It was a combination of getting the assay up and running and getting the protein purified better. And kind of one indication of you'll see if you ever go into or collaborate with drug companies is this kind of Z prime, and this is kind of key for them. They'll kind of go, oh yeah, what's a Z prime on assay? And usually they kind of will say, oh yeah, above 0.6 is good. And really that's just a measure of what the noise is compared to what your signal window is. Right, so let's go on and think about, okay, let's go on and think about studying interactions. Well, it's just A goes to B, we're going to find this binding constant, and that's just really a measure of how much complex is formed over the free amounts of your reactants. Why do you want to do it? You might want to get a KD, you want to say, well, how tightly does something bind? Is it real? Is this a real binding? Is it micromolar, millimolar, nanomolar? You might want to ask how much of the complex is actually formed. Is this relevant to in vivo? Is there environmental changes, pH? Does post-translational modification happen that'll affect it? Which residues are involved? You might want to do mutational analysis. You've got a structure, and you can then say, well, I think this is a real interface, so you can mutate that and see about that a bit later. You can go even deeper and kind of think about the thermodynamics of the interaction. Okay, what about kinetics? Well, the cell's really dynamic. We've got to think of it, being very crowded. And here's another picture from David Gott's cell as well. And so that each individual rates here, each individual binding constant is then made up of individual rate constants of how fast things will come together and how fast they will fall apart. And so looking at the kinetics is really asking what about the dynamics of what's actually happening. So it's crucial to think that not all reactions go to equilibrium. So you may want to ask, what's the rate determining the process? It's like multi-step. Again, you're asking how much complex is formed. And again, go into depth to say how much mutations are affecting this dynamics. And you may then want to look at the heterogeneity of the system using single molecules, which Chris will talk about in a minute or two. Okay, first off, you kind of go, so many different instrumentation that we've got in biophysics. What signal are you going to use? We've got, and really it really depends upon two things. One is, what your question is, and two, how much material do you actually have? So let's kind of work through those things. If you just want to know the affinity and the stoichiometry, and you've got really good samples and good concentrations, best thing, ITC, gold standard, AUC, even, if you've got enough, you want to look at the kinetics, and there's the fluorescence change, you can do fluorescence.flow. If one binding partner is peptide or DNA, you can get it labeled and get it synthesized as labeled, or you could label your partner protein with a fluorophore itself. You could try fluorescence polarisation, microscale thermophoresis, or FRET. So say you understand the dynamics. Well, again, if you've got good volumes and concentrations of one partner, the other one, how could you attach it to the surface via fusion tag? You have a look at biosensors, got different flavors here of SPR, BLI, and switch sense down DNA non-levers. And if you've got a DNA and RNA, you could go again for the switch sense, which is DNA non-levers, or surface plasmon resonance, we'll see is a new kit out recently. So, okay, this is, again, these are kind of looking at things in bulk. If you want to look at the heterogeneity of things, then you need to look at the single molecule level, and Chris will talk about that at the end. See, now you're not wearing the shirt today. Okay, let's go into depth. Okay, ITC, isothermal titration calorimetry. What does it mean? Well, essentially, you've got, it measures heat, it's a calorimeter, and it's going to keep the temperature constant. You're going to react two things together, and it's going to keep that chamber kind of the same. So what you do is you have a sample in the cell here, and you inject in, here's a little injector, which is in this automatic one as well. You just inject into the other sample and you stir it rapidly. And what you get out is you get binding constants, you get stoichiometries, and you measure the delta H, which is the enthalpy, and you can then derive what the entropy is. So we must remember that in biological interactions, we've got bonds being formed here, and we could have conformational changes happening, so that could change then the entropy, and also you get water being released because they're bound onto the surface. When they bind together, water's excluded, and you get these extra waters kind of coming out into the bulk. And then we're going to then measure this equilibrium binding constant here. And then that's related to the actual energy of the interaction with this equation here. Right. So entropy and enthalpy, what drives reactions? So one is telling you the entropy, or enthalpy is then telling you about bond formation and bond breakage, and then the entropy is telling you about how energy is distributed within that entire system. So here's an example of DNA binders. One's a groove binder, and then one's an intercalator, so it goes in between the bases. And here's the energy here, the temperature in Kelvin, then multiplied by the change in entropy. And then on the y-axis here is the enthalpy. You can see they're quite different. So the free energy related to the equilibrium, that's just related to this free energy. And of course, this is something that you probably saw way back when you were in chemistry or whatever in high school. And actually, this instrument actually measures this directly. And what you can see that for the equilibrium to form the products, this needs to be negative. So this term here needs to be negative. So if we have a look at the groove binders here, then we can see that actually the enthalpy is actually just favorable, it's positive, but the entropy change is actually negative. So that's going to drive that reaction here. Conversely, then when you get intercalation, you're going to get lots of bond forming. So you've got lots of the delta H is very negative, but the entropy then change is actually opposing that kind of change. Right. So I just that. So here's what an experiment actually looks like. So you've got a solution in a cell, and you're going to do lots of little injections. Here's all the injections, one after the other, and you get a change then in the heat. And there's a power change in the instrument then to keep the temperature the same. So this is all these little spikes. And you integrate the heats, and then this will then calculate things into this again is the temperature change per mole of injectant, and then this is a molar ratio of what you've injected in over what's in the cell. You get these characteristic curves. So here's a reaction, and here's just whatever you're injecting into buffer, and you're not seeing any heat at all. So what we have to do then is just baseline correct, buffer control, and we get these characteristic then corrected data. So this tells you what the enthalpy change is. You can see where the midpoint is, and that tells you what the stoichiometry is. And then by just doing linear least squares fitting, you can get the stoichiometry, the association constant, which is just the inverse of the association constants, delta H, and then you calculate the values of the energy and the entropy. Right. So how much do you need? It's only got 200 microliter cells, so that sounds quite doable. But you actually need about 350 to actually fill the cell and put the injection in. And you need relatively good amounts of sample because you're going to measure these very, very small heats changing. So the lowest you can probably go down to about 5 micromolar. But how much, we'll just see in a second, you actually need. And in the injectant, you then need 70 microliters. In the auto, you need about 110. And you need a lot more concentration compared to what's in the cell. And which way do you do it? Ideally, you do it both ways. But one problem could be is that you can't get one of the components up to high enough concentrations, you get aggregation. And that can limit your choice. Okay, so how much do you need to do? Well, you need to then get a high enough concentration in the cell to get these kind of characteristic, nice kind of shaped isotherms. And what determines the shape is how much of the concentration in the cell is above the KD. Because you're going to do a stoichiometric titration. And so this is kind of an optimal range. And in the syringe, then you need 10 to 15 times above that. So as you can see, it's going to be a bit sample hungry, but you get good, very, very good data. And of course, it's in solution. So yeah, so it measures the heat of the process, the thermodynamics. You can do anything with it, and we'll see later on what you can do. You know, do conformations, turnover catalysis. Okay, samples can be crude. It's labeled fluorescent solution. You don't need to do much assay development. You can take your sample out again at the end. And you can get kinetic parameters as well, which you can't really go into. Protonation change, oligomeric dissociation. Okay, the disadvantages. Okay, you see everything. So if you don't have buffers matched, you'll see things. If you've got an additive in one of your solutions like DMSO, you'll see that kind of muddying everything. It's very sample hungry. You need to be very, very careful of the controls. Sample solubility, of course, can be an issue. And also you can get protein flocculation because when it injects in, it's stirring as well at 700 rotations per minute. And some proteins don't like that. If you've ever made a meringue with egg white, essentially that's what you can end up doing. So it's not obviously, and these very accurate concentrations. Right. Okay, and you need to avoid kind of buffer mismatches. Okay, here's something in action. And this was then a group solved this crystal structure. And of course, is it just a packing artifact? You've got a crystal, so you packed everything together. Is this actually a real? So whether these two mutations then are binding onto this called a coil. And so what they made is made mutations across the interface. And you can see here that in the wild type, you get nice heaps here. And then when you make the mutations on one interface, destroys the interaction. If you then mutate on the other interface, again, you can destroy the interaction. So that validated that. So here's something, as I said, you can measure all the heats here, something a bit wacky. This is from Tomas group. And they're looking at these kind of bacteria, which then have these coats. And one of these coats, then they could show that what it was doing was kind of taking in ammonia. So what Chris did was we put the actual whole bacteria itself into the cell and then injected in some ammonium ions. And you can see there was heat being released. We're not actually fitting this data. But if we have to destroy the coat protein by then putting in EGTA, you can see that then you actually destroyed that reaction there. This is kind of S layer. Then you put in the EGTA and you kind of get gaps and then destroy it eventually. Okay, so that's that. Another way to kind of look at interactions then, we kind of met this yesterday, just slow speed sedimentation, equilibrium AUC. And then this was a case of a very, very weak interaction. This is from the von Bruegel group. And we've got this crystal structure. Again, is this real or not? You could actually mutate this residue here, this interface. And doing free slow speeds, we could then fit actually KDs to this data here, and then find that the wild type binding was a dimer. It's got a very weak interaction, and the mutant here with the mutation here, it went monomeric. So that was very nice. Okay, let's talk about fluorescence. Okay, fluorescence polarization and all the fluorescence things we can do. Old school, we used to do things in cuvettes, and from 70 microliters up to a mil. We have to use monochromators, and we could take it. We take slowly to just inject in. But nowadays, most people then use plate-based plate readers here in 4 well or 96 well. You've got these kind of volumes, and you can go down much lower. It uses optical modules, so it's not at all as flexible as using perimeter. It's very, very quick. One disadvantage is you can't really get precise control of the temperature. So if you need to kind of go colder, then you have to go into this. So let's think about a very common technique we use to look for assays is fluorescence polarization. And essentially what it does is it just measures how fast something is tumbling in the cell. So let's see. We have something, in our cuvette or in our plate, and we're going to put vertically polarized light across it. And because this is tumbling very, very, very fast, then there's a certain time called the fluorescence lifetime when the light's taken in to when it's emitted. And when it's actually emitted, then that could just be in any orientation at all. And so we could say then that the light becomes depolarized here. And so that kind of tumbling rate depends, of course, on its mass. So then what happens if we then inject in, say, something that binds a protein that's kind of labeled with this fluorophore, and then we'll get much lower tumbling. And so really it tumbles so slowly that when the light kind of gets re-emitted again, it's pretty much in the majority of it might be in the same orientation as the incident light here. And so what we see is when we do a titration, we can see that then the polarization is the same as the incident light, goes up, and the polarization that's in the over orientation then goes down. And then we calculate either the polarization or the anisotropy. And that's just essentially a ratio of a difference over the sum of the total light. Anisotropy here, you'll notice it's got this factor here, this is 2, and this is actually the correct thing you need to fit rather than polarization. But polarization is used quite a lot. Right, this is kind of the data you get. This is in log view here. We can kind of see nice kind of unbound and then bind. So really the polarization and anisotropy is really an expression of the changing intensities due to the change in the tumbling rate, and then hence the mass. So again, we label the small components, and we're going to put it at about the same concentration as the KD, around or below it. We're going to put this constant concentration in a 96 well plate or a 304 well plate, and then we're going to add 1 to 1 of a protein, the binding partner, then from low all the way to high. Put in the plate reader. We're going to calibrate this plate reader. This is one crucial thing. The plate reader downstairs is a Ferrostar, and it has just a kind of set low polarization value, which is there for when you're doing drug screening, because drugs will tend to tumble very, very fast. So you need to calibrate what your labeled component is. We can do that in the perimeter. So you can kind of set what the baseline value is. Okay, we're going to put this into the plate reader. Concentration here. And then we're going to get, this is what the bind is, that's what the unbind is. And then in the middle, this is going to be a function of how much of the total ligand is then in this complex, because this is the labeled thing you're going to actually look at the signal of. And because you're going to titrate this ligand into the complex, we have to use what's termed a ligand depletion model. So we need to know exactly what the concentration of the labeled ligand is. Right, okay, for good data, good buffers, good samples. If you get sticking, put a bit of detergent. Okay, make sure you've calibrated what your F0 is. is there too little light? Because you're looking at just the ratio of intensities, you make sure you've got enough light in there. Make sure you don't have too much, you're not saturating. If you get aggregation, sometimes when you get up to the high point protein concentration here, you kind of start to see your protein aggregating, and then you'll see a large increase in the light intensity, and that's just through the light scattering. But of course, sometimes you actually do get a change in the light scattering that actually matches this titration, and that's because when the partner binds, you get a change in the fluorescence of the fluorophore, but we can actually correct for that. Okay. And do you have enough concentration rings down to the bottom and up to the top to get this kind of nice thing? And again, do you have things in equilibrium? If you set it all up, put it in the plate reader, it takes minutes to do. But you could just say, well, I'll just come back, wait half an hour, put it back in again, leave it overnight. Do I get the same result? Right. Okay, here's an example here. It's a really nice example here from the Barford group, where we came across this complex yesterday. We've got a MAD homodimer, which has potentially two binding partners to this holomer. So does it bind like this, but one binds like this, or is it CDC20 bind like this, or is there a mixture, is it a hetero complex or not? So we then have got labeled then BUB1, titrate in MAD, you can see it's a nice binding here. So then what happens then when we put in CDC20 that's unlabeled? So what we'd expect then, if we're going to form this kind of complex here, then we should displace the labeled one. So the polarization should go down, but instead the polarization goes up, which actually means we've got a heterocomplex. So even though this is a homodimer, it's got two different binding sites. And we could show that again by then preforming the complex and showing that we get the same change in polarization, which means we get the same resultant mass of complex. Okay, so let's think about another one here, which is an technique called microscale thermophoresis. You probably haven't come across this. It's kind of a physical technique that's been known for about 100 years. But the people behind nanotemper have exploited it in this scale. And essentially what they do is it's about the movement under heat. So if we just click onto this. So what they're doing is they are shining an IR laser. And here, then molecules are then moving away. And this is called positive thermophoresis. And in this other one, then the shining a light here. And you can see then the movement of these particles are going towards where the hot spot is. So again, you need to label one component. Similar kind of setup to the present polarization. The nice thing is it's in capillaries, and these are actually set in this holder, which actually matches a 384 well plate. You put it in the MST, and this is the kind of figures you actually get. So you get here, the IR laser goes on, you get this temperature-related intensity change here in the fluorophore, and then you get MST here, and then you turn the IRF laser off. And so you can see movement of this fluorescence away from kind of the imaging spot. And if you just take this data here and this data here, and then get the ratio of the hot and cold, you can then fit. Here's baseline, and here's the kind of the binding. And you need to label with these two labels here, and it's at least 10 microliters a sample. So the advantages are, well, because it relies, this change in MST depends upon changes in hydration or size or hydration shell, then lots of different biomolecules will actually work in this. And you can also then measure these two modes, which is MST or this temperature-related intensity change. And you can look at really, really tight binders with the pico reds, with 647. It's very straightforward. The actual running software is really, really straightforward, easy. It's got a QC. I'll actually tell you, well, is this going to work or not? And here's an example here where it kind of goes, well, let's just do 3 samples bound and unbound. Well, this looks pretty good. If you put the ligand in, we got a nice change. And here it's going, wow, this is a bit scattery, and it'll highlight things to say, signal to noise too low. Let's reconfigure. Okay, the disadvantage is you need to label something, but labeling techniques now are pretty straightforward. It is capillary based, so you've got a lot of glass surface. You could actually get like binding onto one, but you can kind of change the buffer out of detergent. One kind of disadvantage as well is that sometimes you just don't get any signal. We don't know why. I mean, it may be that sometimes things just oppose each other. You might get a change in the hydration shell, but the change in the side, the charge opposes that. But you just have to try it and see. Okay, here's an example then. John and Donald. And so they've got this crystal structure here. And there's a hedge group. And okay, yeah, it wasn't the most highly resolved one. So, but they could fit in structures in. And so the question is as well, actually, is this real or is this not? And they could then just kind of see that actually this complex would actually form. And they went on to do other ones as well to show that this was how the complex was arranged. Right. Okay, so let's think about switch, and let's think about kinetics. Well, okay, equilibrium, we're just really counting what's free and what's bound. Kinetics, we actually want to look at the rates that are coming on and off. So we want to see what the on rate is on, and that is then related to the concentration. So you can imagine if you have more concentration of one reactant, you're actually going to get a faster observed rate. Okay, conversely, with dissociation, you're just seeing what's falling apart. So it doesn't really matter what the concentration is, you're just seeing something coming off. Okay, and that's going to be related to the half-time of the complex. So the important thing is to realize is that you might have an affinity of nanomolar or micromolar, but not all binding is the same. Some can be made up of very fast on, very fast off. Because it's just, the KD is just a ratio of these two values, the fast range of dynamics can give you the same binding constant. Right. And of course, when you get to equilibrium, the on and the off rates are going to be exactly the same, and you're going to get no change in the signal. But even though you're getting no change in the signal, things are coming on and coming off. Right, so let's think about how we're going to do kinetics. Well, nowadays we're going to do things with biosensors. We've got 3 different biosensors. We've got SPR, an octet, and we've got this DNA nanolevers. And when we kind of say a biosensor, what we mean is there's a macromolecule called the ligand. We're going to attach onto a surface, and then we're going to flow an analyte across that. And these all use different physical properties to changes to then measure the dissociation. They've also got different surfaces, and come in different formats. So with Bacro, you get a little chip, and there's the little active surface there. With octet, you get these racks of tips, and the tips there are where the active surface is. And then again, in nanolevers here, we've got a chip again. You've got flow cells here, and you've got electrodes going into these little spots here. So on Bacro, there's the surface, gold surface that's been clamped into a flow channel, and you're looking at changes here of the resonance. Here, you're dipping your sensor into a 96 or 4 well plate and swirling it around. And then in the DNA nanolevers, you've got these little DNA nanolevers here where one has got a fluorophore on it. Right. So the output here is resonance units. Here is this interferometry pattern shift. And here you've got switching speed. So how do you get things on the surface? Well, the easiest way, just covalently couple. You take what your ligand is, and you can just cross-link it onto the surface. It's permanent, and you flow across your analyte. Of course, one disadvantage here is that, okay, you're going to have to do multiple cycles. You're going to do multiple concentrations. Does this analyte kind of fall off again? Or you're going to have to find conditions to regenerate the surface. To kind of get away of that, you can actually have a capture molecule instead. So where it'll capture a ligand, you can do your analysis, at certain concentration, and then you can strip the ligand off and then and then go for the entire cycle again with a new analyte. So these kind of affinity captures are things like GST or his-tagged proteins or strep-tagged proteins. Obviously, biotin-related proteins you can attach them to a streptavidin chip. They're not regenerative at all. Once they go on, they don't come off. There's lots of use of all these kind of assays with antibodies, especially in biopharma. So there's a huge range of ways you can use antibodies to bind onto the chip. Okay, with DNA nanolevers, it's slightly different. You've got the single-stranded DNA attached to a surface. It's got a fluorophore here. And you need then to get the complementary strand to this, attach this onto your ligand by crosslinking, and you can then just flow this across. It will allele, form a double helix, and you can then switch. And then look at your interaction. And at the end of the time, you can just strip the strand off, go back to here, and go all the way through again. And Biocor have, with Cytiva, then have kind of done something similar now, where they've got this capped chip, which recently come out, and it's got a DNA then attached on the chip surface. And again, you get a ligand with complementary sequence here. You can bind on and then look at the analyte as well. So if you've got DNA or RNA, then obviously the best thing to do is actually use the switch sense. Okay, so what it does is it's got an alternating voltage, and the little DNA nanolever is quite stiff. It then gets attracted then to the surface. It's called surface, and gets quenched. If something binds, then you're going to slow this kind of movement down until it comes off. And because you've got a complementary strand here, you can build anything you like on top of it, single-stranded RNA. And in the new instruments, you can actually then build up even to big DNA origami ligands. So what kind of thing are you going to get out? Well, this is kind of a standard kind of what you say is sensogram. You're going to have a baseline. You're going to have something kinetics here happening. It may be that you'll reach equilibrium, and then it's going to stop flowing in the analyte, and you're going to start flowing in buffer, and then you'll start to get dissociation back to the baseline here. You've got these units, SPR units, it's related to the amount of protein bound per millimeter squared. And one resonance unit then is what the angle of deflection, which we didn't really go into. BLI, this is an interference pattern, it's not size. So it's just this wavelength shift. And switch sense, it's a rate of fluorescence recovery. It's just the dynamics related to the dynamics of the switching. So these are the two kind of extremes of data that you'll actually get out, and you'll see in any kind of papers. And we're just illustrating this with some SPR data. So it's fast on, fast off. You get to equilibrium in the center here. Here, you're not getting to equilibrium, it's not flattening out. And it may take a very, 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 very long time to actually get flattened out. So you just don't have that kind of time to run this assay. So you've got kinetics kind of going on and coming off. Easy just to measure this. You just measure it with the equilibrium position against concentrations. This is increasing concentration. Fit it, comes to concentration, you get a nice curve. And then you just fit it to this equation here. And where the dissociation constant is just the 1 over the dissociation constant. Okay, kinetics is a bit more involved because, again, you've got increase in concentration. You've got a dissociation happening here. Now, at all these different concentrations, you're going to end up with the same dissociation constant, right? It's always going to be the same. And these are just simple exponentials. On the other side, though, it's going to be a bit more complicated because what's happening here is that things are going on and coming off and coming on, coming off and coming off. So you need to be able to know what your dissociation constant to be able to fit this accurately here. Right, and we can fit it, so I'm not going to the equations. And essentially then you're going to end up with getting the dissociation rate constant, the association rate constant, you can work out what your binding constant is. Right, and what's the issues? Well, it's cost is the first thing. You're going to buy these chips, you're going to buy the rack of sensors, they all cost money. Obviously, you're going to attach something onto the surface. How is that really going to affect the binding? I mean, you may have steric hindrance, say, you've got something that's bound onto a surface, your partner comes in, and how you've actually bound it onto the surface may affect how it actually gets in. Non-specific binding, this is the killer. You've got a lovely surface, you're going to flow your protein across, you're going to have reference channels, and then sometimes what you find is that your protein of interest just binds like anything to the surface. So you're looking at a very, very small, sometimes you can get more interaction on the reference surface and you can't get on the active surface because in some way you've actually passivated the active surface and you can actually then get a negative results, and that's not good. So there's ways around that. Biocore is very sensitive to refractive index change, so you need to match buffers very well. Switch sense is very salt sensitive. Now there's real limits to how fast you can detect. There's a rate that the instrument will actually measure the data. If you've got a very, very fast rate, it just may not be able to get there. And obviously you can't run these experiments for forever, because if you're going to do multiple concentrations, you can't kind of have each one taking 5 hours. And so really fast rates, difficult because the instrument doesn't take enough data to actually get there. You get these kind of step functions. So when you have a look at that kind of data, which was equilibrium, which is going to step, so there is kinetics in there happening, because obviously things are binding, but the instrument can't measure that fast. It's just seeing a step. Also very, very slow rates. So if you have something that's like 1 × 10⁻⁵ per second in dissociation, then that's going to take a very long time to even get to the half point of dissociation. So one of the issues is really when you see very, very slow rates, is just be very cautious about how it can fit. Because the nice thing about the switch sense is it's got a peristaltic pump and it can just keep on going. It can do very, very long, even overnight dissociations. As I kind of said, then the fitting high affinity ones can be quite difficult. The slow rates can be difficult to analyze as well. And sometimes the computer will actually fit it. And it will say, oh yeah, it's 10⁻⁹ per second. You're going, that's basically a flat line. So just be very cautious when you get very, very slow ones. So best results, there's reference channels in the Biocore, and sense you can use sensors which are on address, which are ligands. And in the switch sense, you can do red and green nanolevers, or subtract, have a good adequate concentration range. So here's an example. This is from the Barford group. And they wanted to look at then the activation of this E3 colon ligase called the APCC, which is very important in the mitotic checkpoint. And they wanted to see then, well, what happened when this activator CDH1 kind of bind? How did that affect then it binding on its E2 ligase? So the biotinolated this whole, I think is it 18 kind of subunits. You couldn't really put that onto the chip surface, because what would happen? Just things might fall apart. So instead, then what we did was then biotinolate then the EH2. That was quite small. You're going to put across something very big, so you're going to get a nice signal. And this is what we could do. It was actually kind of some kind of ideal data, because we could actually fit both the kinetics, the on and the off. It was all getting to equilibrium, and so we could fit the equilibrium data as well. And what we found then was the on and the off rates, then we could work out what the equilibrium is, and then from the kinetics and the equilibrium, pretty much the same. And then without then the CDH1, you can see that the kinetics slightly, the off rate was definitely quicker, the on rate was a bit slower, and it ended up that the equilibrium then was much weaker. And that's where I'm going to leave it. And then.
Okay, so I'm just going to talk for 5 or 10 minutes about single molecule techniques that we have in the LNB. We're going to do a full lecture to cover this in more detail. So what we have in the LNB, we've talked about mass spectrometry yesterday, which is single molecule. We've got optical tweezers, which it's the commercial version. It's called the C-trap, which some of you might have heard of. We've got a home-built FCS, the fluctuation correlation spectroscopy. And then there's lots of other things around the building, turf-based microscopes and things like this. And also we've got an AFM, atomic force microscope. But the two things I'm going to talk about today are FCS and optical tweezers. To start, we say, why do we measure single molecules? The main reason we do this is because obviously the dynamic world is very different to the static world. So if you do crystallography or you do cryo-EM, you get a snapshot of molecules. Obviously, this can be quite different to how they behave in solution. So single molecule studies allow you to look for rare conformations rather than looking at this ensemble measurement. So you can look at, these averages are all the same for these different distributions, but obviously the behavior is very different between the four distributions. You can also see rare events that are on the pathway, such as here. Another reason you might want to do single molecule experiments is because you might actually want to measure or apply forces, and that's something you can do with the C-trap. Often, actually, one of the main reasons people want to do it is because they actually can't make a lot of protein. So a lot of the things that Stephen talked about, even though they're quite low amounts that are needed, they're still a lot higher than what you can make if you're really making a huge complex. So sometimes it's only, the amount of material you've got forces you to do these type of experiments. So this is the FCS experiment. This was built by a former member of the lab, and he described it as home-built, flexible but quirky. So basically laid out on the sort of breadboard that you see with optics. There's 4 different lasers that he's attached, and the big advantage of this is that it is actually accessible. So you can get into it and you can change any of the dichroic mirrors or filters, and you can make it to fit your experiment. So the way it's built around a standard microscope, and therefore we can use the left channel or the right channel to do different experiments. So we can do here, for instance, you can do fret measurements. Here you can do classic FCS experiments. So very briefly, a FCS experiment involves you have to have a fluorescently labeled molecule at very low concentrations, and then these pass through a confocal spot. Then we collect the light that's emitted. It's an extremely sensitive technique because obviously the molecular confocal volume is extremely small. It's done in solution, so you can use any solution you want, any buffer you want, and it doesn't interact with the surface. People do turf. You can do it on the surface, but this is set up to do it in solution. So there's no physical separation issues, and the spatial resolution is obtained just through the optics. A bit like the dynamic light scattering that was talked about yesterday, this FCS. The FCS data is analysed using the autocorrelation function. So depending on the size of the molecule and how fast it moves through this pinhole, and the concentration of the molecule affects these autocorrelation curves. And then by fitting the curves with an appropriate mathematical model, you can then work out the particle concentration, the mass of the particle, viscosity, and also look at the bound fractions. So that's sort of a standard FCS. What is quite a popular experiment to do is actually to do this cross-correlation of spectroscopy. So that's where you can look at 2 different molecules with 2 different labels. So here, for instance, just a red molecule and a green molecule. If they behave, you get a correlation curve for the green and for the red, but you can see if they're not interacting, so if they're not correlating, when you do the cross-correlation, you get no curve. Whereas if the molecules are interacting, they'll pass through the volume at the same time. So you'll get the independent curves, but you'll also get this cross-correlation curve. So it's a way of looking at that the molecules are actually interacting. You can also get all the same information you get from doing the standard FCS. If you go out the left port, we could look at, you can do fret FCS, so similar to the cross-correlation, except this time you just excite with 1 laser, and obviously the fret microscopy is where one fluorescent molecule is excited, the energy is transferred to the second molecule if it's in close proximity. So it's another way of looking for interactions. Okay, so now I'm going to talk about C-trap. I'll just introduce the C-trap here. So this is an optical tweezer. It's based around basically a far infrared laser that's shined into, focused into the back of an objective. That creates a very steep light gradient, and that light gradient then allows small micron-sized particles to be pulled into the focus. And then using the software, you can then manipulate and move these particles around. So all optical tweezer experiments, or virtually all optical tweezer experiments, are designed around using these sort of 1 micron or 500 nanometer beads as handles. So I'll show you some examples of what I mean. So the C-trap that we have is at the LMB is based around a turf microscope, and it's got a lot of features to it. So the things that it has are it's got 3 laser lines, so you can look at blue, green, and red. Here you can see these are 2 beads with a piece of DNA in between, and there was an atto dye that was labeled, so you can image while manipulating the DNA. Ours also has this IRM imaging, so these are unlabeled microtubules that are bound to the surface. So you can, if you're looking at something in the size of a microtubule, it works particularly well for microtubules, but you can do it obviously label free, or the microtubules come in label free, and then you can introduce other fluorescence. It also has bright field imaging. It's temperature control, so you can heat it up if you need to. And obviously it's a C-trap, so you can measure position and apply forces. This system also has a microfluidic chamber, so it has these 5 different, you can put 5 different solutions in, and then you can create flow. So it has a special flow cell that creates these independent channels. So often how experiments are made, you can have the beads flowing in, and you can move to here and capture the beads. Then you can go into your next channel, grab the DNA, and then you can make sure they're connected, go into the next channel, look at protein 1, protein 2, and then image here. So you can design and you can put whatever you want into these syringes and build up your experiment from that. So it's actually quite a powerful addition to this technique. The good thing about the C-trap is that you can do everything simultaneous. So you can collect the fluorescence, the IRM, the bright field, and then apply and measure forces all at once. So it allows you to design quite elaborate experiments, which is something you didn't used to be able to do on sort of home-built systems. This is an example where this optical tweezers was used. Again, this was in the Barford lab. I do a lot of work with them. And this was the idea of this experiment was they had this amazing cryo-EM structure of the microtubules and the kinetical, the outer kinetical, bound to the microtubule. And from this cryo-EM data, they sort of found parts that were interacting, and they wanted to firstly see what forces were generated with this NDC-80, so what force were created. Then they wanted to see what happens when you add the DAM1, this DAM1 ring complex to the microtubule. How does that change the force? And then they made a whole range of mutants of NDC-80 to see how that affected it. So this experiment was designed, you put NDC-80 was bound to the bead, so it's just a single bead experiment. The microtubules were then grown on the surface of the flow cell, and then you can obviously image them with the IRM. And you bring the bead to the tip of the microtubule, you let it bind, or you let it bind to the matrix, and you move it to the tip, and then you pull. And what you get is when it binds to the tip, you pull, you measure this force, and then it hits a rupture force, and then you repeat to get a statistical significant amount of results and measure the forces. And then so this work then complemented their cryo-EM structures and showed that the interfaces that they mutated were in fact making a difference. So there's lots of other applications of the C-trap. There's things that we're doing here where we sort of bending and buckling microtubules. There's a lot of work with the Laurie Passmore's lab, where they're looking at RNA and DNA and a lot of fluorescent imaging. And there's also some experiments looking at the cohesion ring and how this binds to DNA, so that the other side of the kinetical. If you go to the CTRAP website, you can see there's got loads of different examples of the types of experiments to do. So if it's sort of something that you think, working with condensates or, motor proteins, or all sorts of DNA enzymes. But say we'll go into this in more detail in February. And yeah, that's everything I want to say about single molecule things. Okay, good on time.
Well, good morning, everyone. For those that you that do know me, I'm Tomas Morgan. I'm in mass spectrometry here, and I'm essentially here to talk to you about essentially how do things interact? Do things interact? We can use mass spec to sort of look at both of these questions. Yesterday, Kat would have given you a talk on essentially proteomics and how we can look at broad trends across a proteome, and this is really cool. We generate these weird looking plots. I don't do this. I don't really get what they do when they do these, but essentially we can compare different things. So we're always sort of doing a comparison sort of study here. And what's super interesting is we sometimes see things be massively upregulated or downregulated, and that's all well and good, but actually just knowing what one protein is doing is not always that useful to us as scientists. What we want to know is this interacting with other proteins around it? They will likely be pulling stuff with it, but it's not always the case. And sometimes we want to just actually start to understand what's going on here. So is it reacting or interacting with these things, or is it interacting with these things? How do we know? What methods do we have available to us to start to find that out? So this is where sort of structural and proximity proteomics start to come into it. So we start to look at the interactions, and then actually we can start to break this down even further, which I'll get onto at the end of the talk. We can start to look at where things interact, what conformational changes might be occurring with those interactions, essentially how we can piece all of this together. So we're going to keep it really, really broad for the time being, but essentially we can have a protein, this one's in a membrane, and essentially normally when we start to look for potential interactions, we want to add some sort of tag to this, and then we can pull down on that, or if it's not tagged, we can have some sort of IP. But essentially some way of pulling that protein out of a biological matrix. And essentially the first experiment we can do is a really simple one, which we call pull down or IP, depending on what you've got labeled, mass spectrometry. So essentially here, it's really simple, is that you have a tag protein that you pull down upon, usually with some sort of stationary phase bead, and then you wash under native conditions, so sort of in high salt conditions. Things that are very stable stay bound to this protein, so you can work out essentially what proteins are present within your complex, and transient interactions or things that don't interact at all just wash away when we do our bead washing. This is pretty simple. We then just take what's left over at the end of it, we digest it, run it on LCMS. This is really quick, really easy to do, and it tells you really quickly what stable complex proteins you have. So strong interactions are normally preserved in this, and you get an idea of what proteins are interacting with what. What's less good is obviously we lose transient interactions. You've got to do enough washing that you wash away things that aren't actually interacting with it, that just sort of get pulled out by accident. And we don't obviously keep any structural information. The moment we digest this, we haven't done anything to lock in structural information at this step here. So the moment we digest it, we just get peptides and we lose all of our information. So it's a very, very useful technique to quickly understand what might be interacting with your protein of interest, but it doesn't actually generate that much useful information for us. We can go a bit further when it comes to interactions. So we can do a proximity labeling experiment. We call this, the most common one is bio-ID. Now got like turbo ID and even ultra ID techniques, but essentially it's a little bit trickier, but what you need to do is genetically modify your protein on the way to add some sort of biotin ligase. So we add a biotin ligase onto our protein. There's much better. Actually, one of the differences with these new ID techniques is that this system is getting much easier to add. So you add a biotin ligase, you add lots of biotin as well into your cells or whatever sort of biological system you're working with. And then this biotin ligase just whacks biotin onto any protein that comes within the region of this protein. We can then pull down on, we then just put it through a column that can pull out anything that's biotinylated, and then we do mass spec of what's left over. Now, what's cool about this is it can get us a very good idea of everything that's come into the vicinity of this protein. It's incredibly promiscuous, and it's pretty broad in the sense that it's not that selective to what proteins it biotinylates. So we can look for essentially any protein that might be in the rough vicinity. So transient interactions are captured. We can also do this in vivo. Obviously, we're doing this within a cell. We've got this attached to the protein in a cell. The problem is that sometimes it can be very broad. If you've got a protein that's in quite a high traffic area, you can end up having like thousands of proteins that are biotinylated. Obviously, you don't have a complex that probably contains thousands of proteins. So that can be sort of a bit of a con with this method. And it also doesn't get us any structural information. By the end of this experiment, essentially what you get is just a list of proteins that have been biotinylated in some way. So you can actually, obviously, there's lots of variability that can occur there, and we don't have any idea if it interacted a lot, a little. Sometimes you can do some levels of quantitation on it, but not particularly effectively. So essentially, this is another technique that can get you an idea of proximity. These techniques are both useful. They both have pros and cons, but importantly, neither give us structural information. So I'm not saying don't do these. These are really good techniques to understanding how your protein is interacting with other proteins. But if you actually want to start pulling out structural information, sort of the how it interacts rather than just the does it interact, that's something that these other techniques can be useful for, which is hydrogen-deuterium exchange or hydrogen exchange, mass spectrometry, similar to NMR techniques in that what essentially we can look for in this technique is how areas of a protein are changing energetically, conformationally, protection, things like that. That can get us an idea of what's going on there. And crosslinking, which is really gaining a lot of traction at the moment, which is essentially a way of adding covalent linkers into a protein complex to get an idea of what is interacting with what. So this is all pretty simplified, but it gives you a bit of a crash course of the sort of things that we can do. If we know we've got a reasonably stable complex, there's something that you've got tagged up and you've pulled down on a couple of times and you think, okay, I've got a pretty consistent method of generating a nice complex here, whatever method that may be. It can be recombinantly or pulling out cells or whatever. What we can do to this is we can add just a small covalent linker. So it's just like a reactive molecule with two warheads on it, and it reacts to one side and then it attaches to another side. And essentially what this does is add covalent linkers between the protein. Now, what's really useful about this is that when we go ahead and digest this, we end up getting the same peptides that we do in the original pull down experiment, but hopefully, if we've done our experiment correctly, they are left with this small covalent linker. Now, all of a sudden, this completely changes the experiment, right? So instead of losing all of that structural information, we've now retained a heck of a lot of structural information. So when we go through the digestion and LCMS process, we then keep that information. So we've retained structural information. The problem is this is a slightly more complicated experiment. We need to add in a covalent linker at some point. The covalent linker itself not only can, on some occasions, trap states, which is a good thing. It can also disrupt the state. So sometimes you need to do cross-linker titrations, which this is something you might be interested in. We can discuss that when it comes to you planning the experiment. But essentially, we can trap the complex within a state, and then we can start generating information from that. What does this actually look like in practice? Well, we can get, this is a dimer protein, and this is pretty much wrapped around this region of the protein here, so it can give us an idea of what's happening. We've got a tetramer here. I asked this in the last time I gave this lecture. Any idea what this bit could be that isn't interacting with anything? Any ideas? Yes. Exactly, mate. Yeah, spot on. Yeah. This is a G quadruplex. Yeah, this is just sat in the transmembrane domain. It's not interacting with anything. This is a really promiscuous section of the protein that just interacts with everything else. So yeah, that sits in the membrane, doesn't do anything, and then this interacts with everything else. We can actually do this experiment in reverse with a crosslinker that's heavily membrane bound, and we can actually start pulling out information from that region. But in this case, that wasn't what we were interested in. So yeah, this is the transmembrane domain. It doesn't do anything, and then this is interacting with everything else. This is an allotomer. I haven't counted how many proteins. It's a lot of proteins. It does some stuff, but essentially, even for really big complexes, I think this is a kinetical off the top of my head, but I can't remember. But essentially, we can get an idea of when you've got lots and lots of proteins floating around. Obviously, if we were just to do a pull-down experiment here, we'd see all of these proteins, but we'd have very little idea of actually how it's functioning, what's interacting with what's doing what. And in this case, you look at sort of these broad cross-linked regions, and they can give you a really good idea of the strong interactions that are occurring between different proteins. And then when you can map this out in lots of different ways to get you ideas of what's interacting with what. So that's a really useful technique for even from small dimer type complexes all the way up to really large mechanical structures within the proteome. What we can also use is, often what happens is we get handed structures, and these structures can be predicted in loads of different ways. Obviously, AlphaFold is really common, but also old crystallography data and things like that. And what we'll find is we'll find regions where we've got lots and lots of what we call forbidden crosslinks. It's a bit interesting scientifically, because if they're real, then how can they be forbidden? But essentially, if you know that your crosslinker is a certain length and you've got a load of crosslinks that are way above that length, that's suspecting that maybe the structure that you think is giving you that length of crosslinker is maybe incorrect. So instead of it looking something like this, it might look something like this. So it pulls all of those crosslinks within sort of an agreed range. We can use this sort of on steroids instead of looking at this manually and just sort of trying to get an idea of what's going on here. There's papers out. This is from Yuri back in 2023. There's a couple more papers of sort of upgraded algorithms that they've used now called AlphaLink or AlphaLink 2 now. And essentially, if you take an AlphaFold structure and then you restrict where certain areas of that protein can be with cross-linking data, you can get much better agreements to essentially cryo-EM data sets and stuff like that. So this is a really interesting paper. I would definitely recommend if you're really interested in structural analysis and it's hard to get a structure of your protein, especially if you've got intrinsically disordered sections in your protein or protein complex. This can be a really interesting way of essentially reducing the number of predictions AlphaFold is able to just go off and run ahead with, restrict that in, and then get much more interesting and useful predictions of your structure. So that's definitely something to bear in mind. We've been really successful with this with some of the projects in the facility recently, generating large AlphaFold prediction data sets and then putting cross-linking on top, and it really filters down the potential structures to sort of 2 or 3 from potentially sort of 30 or 40 feasible structures. So it's definitely useful. So that's a really top level view of cross-linking. It's definitely worth bearing in mind. If you can get that nice and pure sort of starting point, it's something that we can definitely do quite a lot of. The other one is hydrogen-deuterium exchange. I'm going to preface this by saying this is horrifically simplified. So if you're watching this online later, please don't write me an email. I won't read it. But also, yeah, it's really simplified. So essentially, we've got lots of deuterium or deuterated water. And essentially, the deuterated water on amide bonds can undergo exchange with the protons that we find. So instead of having hydrogen there, we end up with deuterium. And then what we look at is how much deuterium is along the length of the protein. We do this by mass spec in quite a rapid way because of back exchange, but that's not important for our needs right now. But essentially, in this case here, what you'll see is that you've got a region that's stuck out here. That's going to be heavily deuterated, and then the region in the membrane, well, that's not going to be very deuterated. And then we've got these sections of deuteration. This is the most simplified way I can sort of present this, but essentially we get lots of deuterium that happens along the backbone. Now, definitely for any of you here that are interested in sort of ligand binding, small molecule binding, things like that, hydrogen-deuterium exchange can be a really, really effective technique, which is essentially if we add some sort of ligand binding to this section of the protein here, well, that's going to protect the deuterium from getting to it. So essentially, it stabilizes this region of the protein, less hydrogen-deuterium exchange happens, and the amount of deuterium uptake we see in this section drops like that. So essentially, we've got before the addition of the linker or of the ligand, and then after the addition of the ligand. We can then sort of compare these two on a wide scale with lots and lots of different time points, and it can get us an idea of how the essentially the energetic surface of the protein changes over time and with different addition of different ligands. This is, it gives us, I say can give, it does give the most accurate energetic information of your protein. If you're interested in conformational changes and things like that, hydrogen-deuterium exchange is incredibly useful. The problem is it is quite time consuming. You need quite a lot of sample. You need to run lots and lots of injections at lots of different time points to get this to work. And it's also, we're quite limited on protein size, whereas as I was shown in the cross-linking, we can go up to sort of 20, 30, 40 protein complexes. We're sort of limited to about 250 to about 450 kilodaltons on this sort of study. So 450 is pushing it. There's new instrumentation coming out that can push it further, but we don't have any of that yet, sadly. But it's quite a useful experiment. It's something to think about, but definitely don't plan on just running this straight off the bat. Come talk to us and we'll discuss the best ways to run hydrogen-deuterium exchange. It is quite a long process. So essentially, that is just my quick sort of overview of different techniques that we can run, from sort of the proximity type, looking at what's interacting, to looking at like broad proximal labeling, to cross-linking, which, as I said, is really, really picking up interest now, especially if you're working with intrinsically disordered proteins. Definitely consider running this. It can get fed back into almost all of your other data, especially if you're doing lots of cryo-EM. We're working very closely now to feed all of that data back in, and then hydrogen-deuterium exchange as well. So yeah, that's me over and done with. We don't do questions now, do we? We do questions at the end. Yes, perfect. Okay then, well, thank you very much. I'll be back at the end if anyone's got any questions.
Good morning. Over the years, the MRC has invested several £1 million in NMR equipment for putting into the LMB. And we're using it for looking at structure and biomolecular interactions in the solution state. We have an extremely well-equipped NMR facility, and it can be quite daunting to run some of this equipment. So the role of the facility is to help you through that process and offer you the opportunity to run the equipment. You can either run the equipment yourself, if that's what you choose to do, or more commonly, what people would do is come to us and we'll plan the experiments out with you. So you talk to us about exactly what your problem is, what questions are you trying to answer, plan out the experiments, we'll run the data, we'll decide on a plan between us as to how it's interpreted, and then we then make the equipment available to you to get the best out of it. What I want to do today is look at some of the bits of data that we can expect to gain and get from NMR spectroscopy and looking at biomolecular interactions. The first thing that we can expect to do is detect whether or not we have direct binding interactions, particularly in this case, our bread and butter is protein-protein interactions, PPIs. Do we have direct binding interaction between two domains, or do they have a common interface, or do they just co-precipitate as part of the complex? So this can be applied quite easily to either stable or transient complexes, and it's performed in a native-like solution environment. Not only do we identify that there's binding going on, we can also characterize the location of the binding interfaces in there. We can also look at complexes and get high-resolution models of structural complexes to understand molecular recognition events. We have the ability to look at proteins in the solution state, and the large part of the proteome, as anybody who's looked at alpha-fold structures will know, is intrinsically disordered regions. We have the ability to look at binding and residual structure in intrinsically disordered proteins that cryo-EM and crystallography struggle to resolve. Okay. We can look at small molecules binding either as biochemical probes and designing inhibitors so that you can probe your biochemical pathways. You can knock out a particular interaction perhaps, or even in drug lead discovery. Every major pharmaceutical company has had a screening program involving NMR spectroscopy for looking at small molecule libraries. And particularly salient to this is that we have the ability to detect very low binding affinities, even short transient complexes. The other bit about NMR is that the NMR signal is quantitative. So we can use the data to quantify binding affinities, biophysical constants, and kinetic rate constants. So where NMR starts to fall down, where it gets to the end of its scope, is at large molecules. Any molecule that is tumbling very slowly in solution has a degraded signal in the NMR spectrum. So the higher the molecular weight, generally, the worse the quality of the NMR signal. So there has to be, in the end, a limit. And NMR is not best suited to looking at something like this, a multi-protein assembly, or even, in many cases, just the full-length protein on itself, a single full-length protein. NMR is superb if we break that protein down into the individual domain sizes, and we have this ultra-reductionist approach where we take isolated protein domains and look at how they're binding to other protein domains and peptides and small molecules and whatever. So we can then, in this, we can build up a picture by a very reductionist approach of individual pairs of molecules in the solution. As I said before, we can work on transient or stable complexes. A stable complex would be something like this, in a signaling complex in the Wnt pathway that is responsible for embryonic development and tissue homeostasis. But we work as part of an integrated structural biology effort. Our insights complement what we can also see from cryo-EM, crystallography, mass spec, and biophysics. So where does the NMR signal come from, what it's all about? I'm not going to go into huge detail, but there will be a big spread of experience. Some people will never have come across NMR before. So to make sure we're all up to speed on this, the basic principle of electromagnetic induction is that anywhere that you get a movement of charged particles, you have associated with that a local magnetic field. Now that's quite familiar to us on everyday objects like electric motors. We have the current passing through this wire generates a local magnetic field that interacts with the static magnetic field from these bar magnets to drive the motion of the rotor. But this principle of electromagnetic induction applies not just at a macroscopic level like this, it also applies at the subatomic level. So where you have a collection of subatomic charged particles, protons and electrons, inside each atom, you have associated with that a local magnetic field. So every small molecule, every large molecule has a unique landscape of magnetic fields that enable us to gain information from the NMR spectrometer. The idea being each of these atomic nuclei has a north and a south pole, what's referred to as a magnetic dipole. So all of the ones that we're interested in, there are more complicated situations, but the only ones we're interested in have a simple north and south pole. Okay, as we put the molecule into a very powerful external magnetic field, these bar magnets start to spin, and they spin at a characteristic frequency that is dependent upon it's absolutely linearly correlated to the magnitude of the magnetic field that you've got. So each of these different atoms is going to spin at a slightly different rate according to what the local magnetic field is. We can use that information to get us very valuable information on the composition, the functional group chemistry within a molecule. We can also get information about the 3D shape and the time dependence of that 3D shape, the dynamics, and we can use it to look at how it's forming molecular interactions. So what are the sample requirements that we have for NMR? Well, here's the business end of a spectrometer. It's a very simplified one. The ones that we have in our NMR building are cooled down to about minus 250 degrees Kelvin to take out the thermal noise from the circuits. And then just a couple of millimeters away from this business end, the coil, detector coil, is the sample that can be at room temperature. So it's quite an engineering feat to have that kind of temperature gradient so close. But that's the business end, and inside of there goes your sample in a sealed tube. So the technique is non-destructive. We don't even open the tube, so you can get the sample back at the end if you need it. And that sample has to sit inside the coil. Okay, so that's the basic setup. And the concentration range, well, limited detection is somewhere around 20 micromolar. If we go below that, experiment times become so long that the protein may go off. So you have to balance how much sample you put in and how much time you're prepared to have your sample sitting in an experiment. Obviously, if the experiment's going to take 4 or 5 days, then you're going to have a stable system to be working at that kind of concentration. We wouldn't normally recommend that you go above half millimolar. 500 micromolars is a recommended limit. The reason being, once you put proteins in at higher concentration at that, you tend to get aggregation. Okay, so you run the risk of putting your sample off if you go very much more than about half millimolar. That's where we recommend you stop. Temperature range, well, anywhere in the sort of normal boiling range for the solvents, but as long as we stay comfortably below 100 degrees, we're absolutely fine in here. We would recommend a physiological ionic strength buffer if you're doing binding interactions. NMR is quite compatible with those buffers. And if you change the ionic strength to something else, you get very weird results in protein-protein binding studies. And we can, if we want to, it's optional that you can put in stabilizers such as DTT or TSEP, or you can degas to slow down the rate at which you get oxidation of thiols, just to increase your sample stability. So the coils in this setup are configured so that we detect any magnetization that is spinning inside that coil. So that is all of the magnetic dipoles from all of the nuclei in the molecule. What we do is at equilibrium, the bulk magnetic alignment is along the vertical axis, and we tip this into an excited state where it's rotating in the horizontal plane. And the coil is set up to detect the oscillation of those currents. This is a cosine wave shown for a single oscillator. In practice, you would have many hundreds, maybe even many thousands of oscillators in there that's superimposed on this pattern. It's not a great format for us to take any information out of the tool, so it's much easier if we convert that time domain by the process of Fourier transformation into a more familiar frequency domain spectrum. Okay, so we have a frequency axis and the intensity of each component. One of the things you can see instantly is the principal advantage that NMR has over all other forms of spectroscopy. You get an individual signal for each atomic nucleus in the molecule. So when you're looking at UV or others, I don't want to knock them, but you have a bulk effect. You're looking at all N to pi star transitions or whatever. In the NMR spectrum, we have an individual signal for each atomic nucleus. So what's the first thing that we can do? The first question that comes along to us is, I don't know my protein's oligomerization state. Is it monomeric? Is it multimeric? Or is it even in a complex? And the first experiment we would recommend is a so-called spin echo. In this process, what we do is we take the magnetization, put it into an excited state. We leave it for a short amount of time, a short relaxation delay, and it relaxes a little bit back towards equilibrium. The rate at which it does that is dependent upon how rapidly the molecule is tumbling in solution. So the larger the molecule, the slower it tumbles in the solution, and the faster it relaxes. Okay. So if we take two of these spectra and we just run them with a slightly different delay period, we can then get, even from those 2 points, we can get an estimate of how rapidly this molecule is relaxing in between these two time periods, and plug the figures in, we get out the correlation where the molecular weight pops out. This is not a mass spectrometer, but I would say it usually gets us to within about 2 or 3 kilodaltons of the desired answer for a monomeric protein. And it's certainly good enough resolution for us to tell what oligomerization state we're in. So we have this as monomer, dimer, trimer. If you get non-integer masses come out, non-integer molar monomer masses, then usually indicates that you've got an aggregation of some sort going on. But it's a very quick, somewhere, there we go. It's a fast and non-destructive measurement. It only takes us about 10 minutes to do, and we don't have to do any preparation of the protein in terms of adding isotopic labels or whatever. Okay, that's the first bit. So other than that, there's actually very little that we can do with a spectrum in this format because of the amount of overlap of the different signals that are in there. So what we want to do is move into a different spectrum format in which we can resolve all of those peaks. So all I've done in this case is actually exactly the same spectrum, but all we've done is separate these signals again in the indirect dimension here, the vertical dimension, according to the frequency of its directly attached nitrogen atom. So it only correlates one peak for each directly bonded NH pair. That itself is also not a great representation to an easy representation on the eye. So universally, the convention is to depict these graphs as contour maps in which we can now resolve the individual spots. Each of these spots corresponds to, originates from one individual amino acid, and the intensity of the spot is depicted as contours. So this is the basic NMR experiment, the core fingerprint of protein NMR. And what it's showing us is the frequency of the M15 correlated with the frequency of its directly bonded H. But importantly, in order to get this, in order to bring nitrogen into play, we have to label the molecule with N15. N15 is it's an NMR active isotope, but it's present at very low natural abundance. So the only way we can run experiments like this is to grow your protein, for example, expressed in E. coli. We can grow the sample on a minimal medium in which M15 ammonium chloride is the only source of nitrogen. What it gives us is one spot for every individual amino acid. And so we have each of the sites in this case, we have 200 different sites in this protein, and we got roughly 200 different spots. The exact pattern of what these are is not that important. It tells us a little bit of information, but in fact, it's impossible at the moment for anybody to predict, based on the structure of this, what the exact pattern of these peaks will be, because it's dependent upon so many different things. This, all of the proximity, it's the shape of the molecule, it's how it's moving, it's the orientation of side chains and the structure of hydration shells around the surface. So if you change the ionic strength of the buffer, it's going to change your hydration cells, it's going to change the appearance of this. So we can't, with any degree of proper accuracy, predict what this pattern will be. That's not what's important to us. What's important is that it's a handle to tell us how that spot is behaving, that environment, and we can relate that to 200 different sites in the molecule as to what's going on at any one time. That's the power of the HSQC. Okay, so here is how we would use it in practice. Just a standard experiment in which we've got, in the Wnt signaling pathway, we have a PDZ domain from the disheveled 2 protein, and it's binding normally to a C-terminal peptide in the same molecule. So what we can do is we have an overlay of the 2 spectra with and without disheveled C-terminal peptide. And what we can do is we can assign which of the spots is which, and therefore plot out the magnitude of the change that we see onto a structure from the PDB of this. And it shows us in this case that absolutely this disheveled C peptide sits in exactly the canonical binding cleft that we would expect for a PDZ domain. In this particular case, then, we were using this to compare how this behaves with the V334E mutant, which was postulated to be a non-binding mutant. V334E is sitting right in the middle of this binding pocket, and it confirms that in the case of the mutant, it doesn't bind to the disheveled C protein. So V334E is a non-binding mutation. An interesting corollary that we can get from this is that we can overlay, in the absence of ligand, the spectra from the wild type and the mutant, and we see that most of the signals in here aren't perturbed at all. So what that's telling us is a great QC check that we have in all of our NMR experiments. And what it's saying is, if that fold were to change, if the wild type fold were to rearrange in any way, even a small amount, we would see large amounts of perturbation in the position of those peaks. So the fact that we don't have widespread perturbation of peaks in there confirms to us that the mutation retains the wild type fold. And in this case, it's a great example to look at the effect of knocking out that particular interaction and what does it do to the rest of the pathway. You can be a little bit more quantitative with the chemical shift mapping. So in this case, if you have this deubiquitinase enzyme called TRABID, and the way that it's interacting with ubiquitin, if you just had one addition of ubiquitin, what you would see is the spectra in blue and red. You wouldn't really know which peak has gone where. If you have an intermediate concentration in there as well, you can immediately see what the trajectory of all of these perturbations are. I say all of the perturbations, some of the peaks aren't perturbed at all. That's showing we're only getting changes at the binding interface, and this is why we're able to identify where the binding interface is. But it gives us an opportunity to plot out where the interaction regions are and to quantify what the binding affinity is by ligand titration. And just by plugging all of these figures into a standard quadratic equation, we can get out the value KD for the binding affinity. And it's one of the gold standard methods for working out KD in tricky situations. The limit of detection for this is approximately, we need enough protein for about 1/5 of KD to see any perturbation at all. And we can measure affinities as weak as 10 millimolar. Remember that KD and affinity go in opposite directions. So a high KD means a very weak affinity, right? And that 10 millimolar is pretty close to non-specific binding. So NMR is sensitive enough to be able to quantify binding for really weak associating systems and for stronger as well. That's the upper limit. There is no lower limit in this case. We don't have to look just at simple two-state systems, two-component systems. This is a case of a real-life study on the protease thrombin from the blood clotting cascade. And it binds to 3 different ligands, and you can map out individually the state and plot. The punchline of this slide is actually quite difficult to see, unfortunately, but it's the color coding of each of these graphs is showing where we have allosteric changes upon binding of a particular ligand. And you can deconvolve that entire pathway in terms of the allosteric changes that take place on binding ligands. So there is a complexity that comes in when we're looking at signals in how NMR signals behave in the case of dynamic equilibria. So if we have a very slow exchange or no exchange situation where we've got, let's say, a 2-state equilibrium, and it can be either due to ligand binding, or it could be different conformational switch states, or chemical exchange, whatever. But we've got essentially a free form and a bound state, and they have different frequencies. In this particular example, I've set them up so that they have equal frequencies. If you change the exchange rate between these, you get a change in the appearance of the spectrum. So for a very rapidly exchanging system, we don't see 2 signals anymore. We see a single signal that is exactly at the midpoint between the two extreme limiting cases. If I then, so what this is derived from is just by, all I've done here for these plots is plot out this expression, and all I've changed between the different plots is the value kx, the exchange rate. And what it drives us from is a situation where we're going on what's referred to as the NMR timescale. Right? Is the exchange slow, fast, or intermediate on the NMR timescale? And it gives us quite different appearances of peaks. If we titrate in ligand, in the fast exchanging case, we got what we just saw in the HSQC. We get a migration of the peak going from, at the limit, it will go from 1 frequency up at 100% occupancy of the protein. It will go up to here, right? But then as we titrate in, that migrates across. In the slow exchange case, we can also get the proportion of each of the species that's present quite easily. But if we were to do fitting on the intermediate exchange rate, we have this process that's referred to as exchange broadening. It does what it says on the tin, right? And in that case, we actually don't see any signal above the noise level of the spectrum. If we were to do any kind of fitting to work out affinities from these kind of spectra, we would need to know the spin frequency of both states, the population of both states, the relaxation rate of both states in the limiting case, right? And the exchange rate. We would have to have accurate values for all of those in order to be able to fit it. So when it gets to intermediate exchange, we're less likely to offer getting binding affinities. Here's an example of what we normally get, a real world sort of case where most of what we see for binding interactions is that we see exchange broadening and the disappearance or bleaching, if you like, of peaks. So this in the black is a spectrum without ligand, and we can add ligand in there and it bleaches out the signals that you get for those that are at an interface where we have intermediate exchange. This also illustrates another point that I wanted to bring in. Do we have to have an HSQC spectrum in which we're correlating N15 and proton? Well, yes, it's preferable because it's the most sensitive and easiest for us to run. But in some cases, it may be better to pick another pair of nuclei to resolve the overlap. And in this case, we've done it for carbonyl carbons. So in order to get that to happen, to bring carbon nuclei into play, again, we have to add that at the time we're making the protein. And we grow the protein on a minimal medium that has N15 ammonium chloride as the sole nitrogen source and C13 glucose as the sole source of carbon. Okay, but what it gives us is the ability to look in this particular case at proline residues, because you may be aware that a proline doesn't have a backbone NH. So when I say on the HSQC, you see a spot for every protein, for every amino acid in the protein, that is every amino acid except proline shows up. Right? In this spectrum, every amino acid shows up. This is just a cutout of the proline region, and it's showing us that we can get resolution in there. There are 10 prolines in this particular sequence, and 5 of them are bleached as we add the ligand. When we look at the sequence here, here's the position of the prolines. We can see that these are the 5 bleached residues of prolines, and the non-bleached ones are in a non-interacting region. So it's proof positive that we have something going on. We can get a high-resolution structure of a complex. The idea is using this program Haddock that's now been around for quite a long time. We can dock small molecules. It has flexible body docking, so we can relax the surfaces so that they kind of meld into each other properly rather than just doing a rigid model and trying to force them together like a Lego brick. This can flex and give a better depiction of the complex, and used as restraints is the chemical shift perturbation. So we've highlighted in orange here those residues that have chemical shift perturbation, and they are also surface exposed, right? Because you'll get some chemical shift perturbation that is from the core, the rearrangement of the core, but that doesn't make a docking surface. So we put in the additional constraint that it has to be more than 50% exposed, surface exposed. In this particular case, we're going to supplement it with some other NMR data. And this is a so-called nosy spectrum. Each of the peaks in this spectrum is what's called an NOE, and it arises only when we have close proximity between 2 spins. So that peak is telling us that a proton at that frequency, right, that's spinning at that frequency, is very close in space to a proton that's spinning at that frequency. And the intensity of the peak is inversely proportional to the distance. So as we get down to really close distances, the peaks get huge, right? So that's an intermolecular ruler. It's called an NOE, or nuclear Overhauser effect. We got an additional frill to put onto this particular experiment, where we put on a couple of what's called half filters. So what we set up is a filter in this case in which we'll reject the signal from any proton that is directly bonded to a C13 nucleus. Okay. On the horizontal axis, we only accept signal from protons that are directly bonded to a C13 nucleus. That might be a little bit difficult to get your head around until you realize the only signals that pass through this filter have originated from the unlabeled peptide and crossed over onto the labeled protein. Okay, so all of these signals are showing close proximity specific contacts that we can identify that are between 2 specific protons across the interface. We then use those as restraints in this flexible docking, and we get out a high resolution model. The reviewer that we sent this paper to originally doubted that this side chain can reach out that far. It said it was much too big a distance, it couldn't possibly happen. We did this study, sent back the manuscript to the same reviewer, who I have to boast, then called it a tour de force. So that was a remarkable turnaround in fate from one of our skeptical reviewers. But it works, and it's a definitive answer of what the complex is. There's another way of looking at proximity in molecules, which is using so-called spin labels. These are paramagnetic spin labels. Now, paramagnetic center is a bit disastrous for NMR signals. Anything within about 30 angstroms or so of a paramagnetic free radical is going to get bleached out by the paramagnetism. So what we can do is experiments in which we attach one of these paramagnetic spin labels onto a protein surface. Anything that is within proximity, within about 20 or 30 angstroms, is going to get bleached out. Now, the control spectrum of this is not with and without label. It's before and after reduction of the free radical to a hydroxylamine. So we get facile attachment of this to cysteine thiols. So you can make single mutations to introduce a cysteine in your molecule and then easily attach this tempo label to it. And then under mild conditions, we reduce it with ascorbate. The detection can be for stable or transient complexes, populated states, and we can look at both intra and intermolecular proximity. So this is rather similar to FRET labels, but in a single label. And here's an example of it from Roger Williams's group, where we're attaching a paramagnetic spin label to ubiquitin, and we're looking at the NMR spectrum for an N15 labeled superdomain. So the only signals that will appear in the NMR spectrum are from the labeled domain. We don't see signals from ubiquitin itself. We only see the influence that ubiquitin is having on the spectrum of SUBA. And when we look at that, this is before and after reduction with ascorbate, and you can see a dramatic change in intensity of some of those peaks. You can quantify that peak height ratio, and from there you can get an idea of where in proximity the surface of the SUBA is to the spin label that's on the ubiquitin. In this particular case, we have made two separate mutations from a K6C to which we attach the spin label and a K48, and comparing those and combining the data, you can get the orientation of ubiquitin in complex with the SUBA domain. We get asked from time to time, what is the molecular weight limit for the upper limit of molecular weight for protein NMR? And the answer is, it's not a hard and fast limit. It's a bit of a black art, and it's dependent upon what your system is. In this particular case, we're looking at a protein that is 175 kilodaltons. Normally, comfortable for NMR is about 30 kilodaltons or below, maybe 40 if you're pushing it. So this is very big in NMR terms. And these signals, these small ones out here, are they are from the protein, but they've been bleached because they're so diminished in size, right? But we can see some of the residual peaks in a complex of this size. And when we add a binding partner, a known binding partner, we can see definitely there is an interaction taking place. When we repeat this with the individual domains, we can actually exactly repeat this with smaller fragments that shows what we see with the smaller fragments is also occurring in the full-length complex. So to say what's the molecular weight limit, well, we're not going to get high resolution data from here, but we can certainly tell you what's going on inside that molecule in the larger state. We can also go to larger systems if we look at methyl groups instead. Now, a lot of the fast relaxation and the diminishing of the quality of the NMR signal, it arises from very fast relaxation that occurs through interactions of protons with each other. And it's very efficient at causing the excited state to relax back very quickly. What we can do to slow that down is we can deuterate the molecule so that it doesn't relax nearly so quickly. And then we can reintroduce labeled amino acids selectively labeled with proton and C13 in the methyl groups. So we add precursors of the biosynthetic precursors into the growth medium that make methyl groups in isoleucine, leucine, and valine. So we can get selectively back protonated resonances that we can then run methyl correlation spectra. This is a proton and methyl carbon on here. We can also do this more simply for methionine just by supplementing the growth medium with methionine that has been labeled with C13 in its see epsilon position. Okay, and then run these experiments referred to as methyl-trosi. They're very similar in format to the HSQCs that we've been seeing, and we can extend the effective limit up to around about 200 kilodaltons. Unfortunately, the bad side of this is to assign these, the only way of doing it on these spectra would be to mutate out and do an alanine replacement and see which of these methyl groups disappears. So it's a difficult assignment, but the information is absolute gold dust for looking at high molecular weight complexes. Why do we want to look at high molecular weight complexes? We shouldn't get too fixated that all we're going to look at is the biological macromolecule. In this case, we're looking at the turnover of substrate. So just a DNA translocase in which we were turning over ATP into ADP, and we can follow time courses for that. We don't have to be fixated on experiments that will look at high molecular weight complexes. We can also do compound libraries and collections of compounds to speed things up and run them as cocktails. Here's some cocktailing of 4 compounds per cocktail that we ran. And we show that when we get a hit, we run the individual compounds, 4 compounds on their own. And we can see in this case, just one of those 4 compounds was able to reproduce all of the changes that we see in there. And it's this compound here. Because we know the assignment of and the identity of each of these peaks, we can then plot the surface, the binding surface, which is shown in red here. Gray is proline residues for which we have no information. But we had perturbation in here, and later when we got a crystal structure of this, eventually it showed that the molecule binds into the target pocket we were after. But NMR have been able to screen quite quickly, 1, whether it was binding, and 2, where does it hit? Does it hit our target pocket or not? Here is an overlay of 5 different NMR spectra, they're reference spectra for 5 small molecule compounds that have drug-like potential. So we got a green, a lilac, a red, orange, and magenta. And when we run the assay for this one, it's an assay referred to as waterlogsy, and it gives us positive peaks for any molecule that is binding to the surface of the protein, and negative peaks for any molecule that isn't. So in this particular case, it's easy to pick out from cocktails of compounds, if you need to do it, detect whether or not it's binding, and then characterize an affinity. One of the things we see as a problem, not just in NMR, but also in ITC, is when we're looking at a binding isotherm like this, we can see that this saturates. Now, as this is plateauing off, is that plateauing because we've saturated the protein, which is what we're after, or is it because we've reached the solubility limit? And this is an example of an alternative assay to the others, where we look at the signals just of a SPI compound. So we're only going to look at the signal of a compound that we know binds in our target pocket. And we use that as a SPI, and we use other molecules in competition binding to displace the SPI. So in this particular experiment, if we have no protein, all of the SPI molecule is in bulk solution, and the peak intensity is as shown here, right? If we put in the protein, we have maximal amount of SPI molecule bound to the protein, and the peak intensity is up here. Now we start adding competitors, fine, and it will displace the, well, we'll get a peak height that quantitatively depicts how much SPI has been depleted, displaced by the competing ligand. When we do a plot of what is the KD we get from ITC and compare it with the KD we get from this assay, and we should see a nice straight line correlation along the diagonal. And what we had in this case was one unknown in there where we have flagged it up as problematic. And it does indeed show, in this case, we haven't had to titrate into the level where we're reaching the solubility limit for the compound, and it's showing out as an outlier in this plot. And indeed, it was genuinely a molecule that was poorly soluble rather than as good a KD as it was apparently showing. So that is a summary of the whole lot. And we've been in this slide before. So we've just looked at the ability to detect complexes, the high resolution structures, the fact that we can apply it to intrinsically disordered proteins, which are becoming more and more of interest in the kind of work that we're asked to do. We can use it for probing cocktails and looking at ligand binding for small molecules. And it's a quantitative technique where we can get binding affinities, rate constants, and equilibrium constants. And that concludes that. I thank you for listening.